This is TWIS. This Week in Science, episode number 930, recorded on June 14th, 2023, the Upright Science Brigade. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with Asgard, Australopithecus, and dead flies. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Artificial intelligence is in the news lately, with worrisome warnings of a future beyond our control. So before anyone panics about our future robot overlords, a little context. AI is nothing more than a collection of computers that can read everything we have ever written on any subject and maybe learn from it and apply it in ways that we might not expect. But what AI does not have is a motive. AI has no interest in humanity, no will or ideology it seeks to impose upon us, and no desire to change the world or profit off of its people. Eventually, if not already, we will be giving goals with human motives to the AI, and given what we know about humans, these goals will likely revolve around lawyers, drugs, and money, or some combination thereof. There are archaic forms of artificial intelligence that already operate in the world today. We call them corporations, governments, and science. These are all forms of an artificial intelligence. As these entities seek to incorporate more powerful AI into their operations, what we will see will be nothing new and nothing to be afraid of that there isn't already good reason to be worried about today. Corporations have tremendous power over resources and leverage over people, historically showing a lack of concern for human health and well-being outside of its own bottom line, and this will influence how corporate-motivated AI treats people. Governments around the world already range from Big Brother authoritarian dictatorships to Big Brother limited freedom democracies. Government-motivated AI will reflect this, making any system harder to challenge from within. And science, which is already awesome, will just get more awesome. The science-motivated AI will accelerate every form of research and give us much to talk about here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back. Yes, we are. Oh my gosh. Who would have expected that? A weekly science podcast coming back again in another week? That's right. We're back to talk about all the science that we think is important and interesting to discuss this week. This is a Big Brother free zone. Just the science. All right, everyone.
Unless you're watching on a social media platform.
Right, I was about to say, we are linked to Facebook, so...
Yeah, no, there are all these caveats, always caveats. Anyway, on the show tonight, I have science about our Asgardian roots, dancing peanuts, and tunnels in your brain. Justin, what did you bring?
Those are all amazing sounding.
They're gonna be.
Let's see, I've got out of Africa earlier than we thought. Maybe. Lucy, the upright Australopithecus. Another reason we need trees, in case we didn't have enough. And why smoking is killing non-smokers at an alarming rate.
Oh, no.
Oh, jeez. All right, Blair, what's in the animal corner?
Oh,
Yeah.
my goodness. I have hot sand. I have death perception. Yes, death. perception.
Not depth.
And
Yeah.
before that, I'm going to have a real quick story about rhino horns.
All right. I'm looking forward to all of this. I can't wait to talk about all these things and even more, because I know there's more in store for everyone. And I hope that you are all looking forward to it too. If you have not yet subscribed to us on the podcast machines, go into the podcast realms. Look for This Week in Science, all places that podcasts are found. TWIS could work too, but This Week in Science is what we're called. You might be able to subscribe to us. We also live stream weekly Wednesdays, 8 p.m. Pacific time. What is it? 5 a.m. Central European time right now. On Thursdays. Anyway.
Thursdays.
Live. YouTube, Facebook, and Twitch. And we would love to see you here in our chat rooms. You can also subscribe to our channels and watch those shows asynchronously if that's what you like. Additionally, we're on Instagram. We're still on the Twitter, even though we're not really on the Twitter. We're on Universidon, if you like the Mastodon thing. And Facebook. I already said Facebook. Just look for twis.org. That's our website. Show notes, all sorts of other interesting things if you need resources. Time for science?
Yes.
Let's do it.
Humans looking into the solar system. That is my first story for the night. We've been peeking at the solar system, and, several years ago, there was this little tiny spacecraft that orbited...
Hayabusa.
Nope.
No?
Orbited Saturn, and it did a death dive into Saturn, and it took all sorts of pretty pictures. Remember the Cassini mission? Cassini, yeah. Took a long time to get to Saturn, and then when it finally got to Saturn, it was there longer. It was a really great mission, did all sorts of stuff, it sent off the Huygens probe to one of the moons of Saturn, like all this great stuff. It is still giving us good information. The data from Cassini is still being analyzed, and researchers who have just published a new paper, detection of phosphates originating from Enceladus's ocean. So this is data coming from the Cassini probe from one of the detectors on the Cassini probe, which is the Cosmic Dust Analyzer, in which it flew through plumes of water, ice, and these plumes that are ejected from Enceladus, and also flew through the rings of Saturn to be able to get an idea of what kind of particles were there in the dust and gas around Saturn in the rings, and also emanating from the moons like Enceladus. And in the process, this information is just now coming to light, which is amazing to me because it's been years since Cassini crashed itself into the surface of Saturn. And researchers...
This is also, by the way, this is part of why we need AI in science. These missions generate so much data that you've got to chop it up into these tiny, small little pieces and hand it off to scientists to analyze. And the entire process is... Our data collection ability is so much greater than our capacity to analyze at this point, that this is why it takes. There's a time delay between collecting a ton of data and
there's a time, there's that time delay, and there's also the publication delay. And so these researchers who were involved in trying to figure out whether or not there are signatures of compounds that are involved in organic life. Carbon we know of as, the basis for organic chemistry, organic biology, chemistry, but there are a number of other elements that are important and also responsible for life. Phosphorus is one of the main key elements for life. And in the dust and gas, they have specifically been able to find the signature of phosphorus emanating from Enceladus. And this is interesting because years and years and years, they've been hypothesizing that the icy crust of Enceladus hides oceans of liquid methane, liquid that may contain amino acids, that may contain other compounds that could support life. Could there be alien life, microbial life, even, or even more, could be little alien octopuses hanging out underneath that icy crust. Who knows? But the question is, how do we find out if it's there? And we have to do these measurements. And so the instrument on Cassini was able to take these measurements, but this model was these researchers said back in 2020, they said, we think that there might be phosphorus on Enceladus. And then they published a paper about their model that came out in 2022. So 2 years later, their paper came out. And now in 2023, they have actual data from Cassini that is actually spectroscopic. They've got scanning electron microscopy related to phosphorus chemistry and molecules. And they're like, yep, there's phosphorus on Enceladus or in Enceladus under that ice crust.
Mm-hmm.
So basically, we need to go to Enceladus, crack it open like an egg, and see if there's life there.
And be sure not to bring any with us, so we accidentally think that that's what we found.
Yes. Yeah.
The thing is, assuming that life always starts the way it does, which 50% chance.
Maybe not.
One of the things that would happen is there's a possibility that we would infect another planet, right? This is
right.
The other idea, though, is that we might hybridize with it, make both stronger. This is how life finds its way around this planet.
Okay, look, your cousin Jeff Goldblum may have said that life finds a way, but in reality, we just want to find out what's out there before we decide whether or not we're going to hybridize it.
That would be preferable. Figure out what the endemic species are first.
Oh, deciding things ahead of time. All this planning.
All this planning. So much planning ahead. Well, this was not ahead of time planning. And in fact, it's one of those wonderful things that comes out of data. So I'm really glad to see Cassini mission data still providing us with lots of information. And then my second story for how we're looking at our solar system, researchers from the University of Copenhagen have just published their study determining, well, it's also in Nature, and it is not necessarily a new idea, but that the Earth might have formed a lot faster than we originally thought it did. So instead of taking 1 billion years to form, or more than 100 million years to form, then this new study suggests that there was a vast accumulation of little tiny millimeter-sized pebbles, according to Martin Bizzarro, professor at Globe Institute, one of the researchers behind the new study. The Earth was formed in just a few million years. And based on their simulations, their findings, they say it appears that the presence of water on Earth is a byproduct of its formation. So it gets past the concept that water on Earth was a chance of impact with comets from the outer solar system and is just a byproduct of the combination of a variety of pebbles, of gravel and stuff that was already in existence in the ring, and that a little tiny conglomeration had more gravitational pull than others and just eventually built up and cleared its orbit, and that is the Earth, and that would have been the way that our Earth formed.
So one of the things that I don't know if this reinforces or is a counterpoint, because like actually could be seen kind of maybe it's both.
Maybe it's both. I don't know.
Itokawa, the pebbly asteroid that Hayabusa sampled long time ago, has been found to have salt crystals that can only form in liquid solution. This is not an outer water source then. What it indicates is all these near Earth objects that we assume to be dry could contain enough water at some point to have been liquid and for the salt formation to have taken place.
Or that it was never liquid, but it's the salts or the combination of molecules. So these compounds, when they come together in a certain way, then you have water within. And we know that there is water in the crust of the Earth, not in like liquid lake water form, but actually tied up in rocks.
Yes.
So, yeah.
But that, I mean, so then it might not need one of those like faraway objects to come and brought water here.
Maybe it's both. Yeah, but
or both would even accelerate everything.
Yeah, perfect. It could be both. It could be all. But this particular study, the theory that they're working on, they were investigating silicon isotope constraints on terrestrial planet accretion. And so this is their way of kind of looking at isotopes of different molecules to be able to compare between 60 different meteorites and planetary bodies and try and like phenotypically, like Blair, the way that we look at like biology and genetic relationships, try and establish that kind of a relationship through chemistry and what we know of how chemical timing works and how that works, to determine how various rocky bodies might have developed into the Earth, which is, I think, a very interesting approach.
Yeah.
Yeah. So it's like, instead of looking as we have at the Earth from an anatomical perspective, which is like what we used to do, is like, oh, this bird has feathers, so it's related to other birds,
Right.
Basic morphology.
Yeah, morphology versus physiology. Yeah. Anyway, interesting stuff. What you got there, Justin?
Oh, that's a great question. Where did I start off? Oh, yeah, this is a pretty big story. Let me see if I can find it. Yeah, never let the thing you do not see be the evidence that they are not there.
Fair. Yes.
So, this is sediment layers of a cave in northern Laos containing some human skeletal remains. Has been dated to between 68 and 86,000 years ago. Now, this is, and along with evidence that human presence in the region was lasting, I guess, for like 56,000 years. So, in some ways, this find is unexpected, because this would be like the oldest site found. And other ways, it's just what you might expect. So, it's unexpected in that there's the oldest modern human site found in Southeast Asia by about 40,000 years. And so falls outside of our confirmed knowledge of human migration and occupation in the region. It is, I think, expected, as we know that people reached Australia between 50 and 65,000 years ago. And so we should expect there to be settlement sites somewhere between Africa and Australia that predate Australia. That, or people just were like, we're on the move, we're not going to stop until we get to Australia.
There should be some evidence of that, though, still.
Yeah. So there should be places in between that are older. And there are some sites in maybe India that seem to be older of current modern humans. So there's that beginning. But then, this is quite an astounding find. The Northern Laos find suggests that humans have had other strategies, because part of also why we think we haven't seen any of those sites over those incremental steps as we expanded out of Africa is because there's a previous assumption that human ancestors only traveled along the coastlines. And so those ancient coastlines are now out to sea and underwater due to sea level rise, obscuring any evidence. This is sort of highland inland, and in fact, it actually makes quite a bit of sense, because the early explorers they think in this region that found this cave were likely following inland rivers. That's such a better strategy, it seems, when traveling, following a river than following a coastline, because there's fresh water there. Wherever as long as you're near a river, you've got fresh water, which seems like that's like a tough one to wander away from if you're an early hominin without at least if you don't have a decent canteen, right? Or something to carry water in. The ocean doesn't help with that at all. So why would anybody willingly follow a coastline? You can't drink that water. That's no good. So, anyway, there is no DNA from any of these fossils. And there's no direct dating of the fossils because Laos has prohibited the destructive analysis of any human fossils that are found in any sites there. Which is great.
That's good. Yeah.
On one level. On the other, we need the DNA. So the researchers used workarounds to date bovine teeth that were found in the similar layers. They used uranium series dating on a buried stalactite fragment, which is a very accurate sort of atomic clock way of doing things. The site is also close to Cobra Cave, which I think looks like a cobra from the outside. I don't think it means that it had cobras in it. What they did find in that cave was Denisovan remains.
And snakes.
Previously. Well, so that site, though, is previous site that was found is 70,000 years older than they think this site is. So it doesn't mean that the humans and Denisovans met there. But, at some point, we know Denisovans and current modern humans had hybridization events throughout Asia, and especially Southeast Asia. And so somewhere in that overlap of the last of the Denisovans and the early explorers to Southeast Asia, there were interactions that took place. This now being the oldest human site in Southeast Asia gives a great possibility that these early modern humans may have been. Now, they say they don't think there's any hybridization in the fossils that they found thus far. It doesn't look like they were already hybridized with anything. They just look like the current modern humans of the day. And likely they're speculating that these aren't the direct ancestors of Southeast Asia. Part of the problem, of course, is they don't have the DNA. So I think any speculation, one way or the other, shouldn't happen. Really. Like, get your evidence
that's a very
first.
fair. I mean, you've got the stuff there, but you don't have the DNA. So
hopefully in 10 or 20 years, there'll be some new technology where they can scan DNA without destroying the material, and they'll be able to figure it out.
There is. That's the next step. They're going to go through and apply environmental DNA techniques to the soil to hopefully capture enough of these early humans somewhere in that collection to get a better picture. They have bones. I wish they would just try just for this. Just make an exception, mouse. Let them do this one thing to try to see.
Okay. But environmental DNA, I understand it for doing here and now kind of assessments, but seriously, like to make sure that you are getting the DNA of a long extinct hominid that
is very undisturbed soil. Yeah.
Very undisturbed soil.
These wouldn't be extinct.
And not,
these would be current modern humans, but from 70.
Right, but then how do you know, like, honestly, I really want to talk to these people about how they're going to keep it clean. How do they keep their human DNA out of the mix so that they absolutely know there's no contamination?
Right. How is not every person who's been in that site since then now contaminated the environmental DNA? Yeah.
How many people have walked into and out of this beautiful cave? Because it's a gorgeous cave. I want to live there.
So I think there might be, I mean, I don't know exactly the technique that they use. I think everyone who's working in this needs to have their DNA sequenced so they can be compared to these. But we were talking about the proteomics. Just in proteomics analysis, they were able to back engineer the individuals that they came from.
Yep.
So there are enough unique signatures in tiny segments of DNA that will only have come from certain individuals. So, and it is tying exceedingly small things, strings of information together, which, again, kind of an important task that AI can accomplish that humans have a harder time.
As long as it's not hallucinating, but yes.
There is an asterisk here, as there are claims of older modern human remains found in China. However, whenever those have been investigated by researchers and scientists who are not from China, they turn out to be inaccurately dated, not human, or have genetic analysis that matches so well with current modern Chinese inhabitants of the region that it would have to come from a current modern Chinese inhabitant of the region.
Right.
To Blair's point. So asterisk, not asterisk. That can happen. It does happen. And it has happened quite a bit. I think actually, was it Svante Pabo's first work in ancient Egyptian DNA, which was a PhD thesis project, turned out to be contaminated by researchers.
Yes.
And
he's gone on to do really amazing work, but I think he learned his lesson. Yeah. He's like,
didn't go like, I failed at science. I will now no. Instead, he's made
it's how do I
contributions to science. Yeah.
Let's fix the problem of contamination and let's move forward. But it's a huge issue especially when you're dealing with this kind of genetic human ancestry genetic data. You just have to make sure that it's not contaminated. And so I want the eDNA thing to work.
Yeah.
But
this would be a good guess. We should get somebody on here who can break down how come it works.
Break it down. Speaking of how things work, Blair, you want to talk about not bones, but rhino horns?
Yes, rhino horns. Just like our fingernails made out of keratin. They are sought after for many reasons, and because of that, every species of rhino currently on the planet is endangered. And so this is a study from Université of Neuchâtel, which is in Switzerland. It's a French-speaking area of Switzerland. They wanted to look at some of the impacts on some rhinos who are currently protected. So this is black rhinos and white rhinos they were looking at. And there's been a variety of ways to try to protect them. The most common is that they're armed guards, that their entire job is guarding a specific rhino or group of rhinos. But more recently, there's been an attempt to remove their horns so that poachers have no reason to kill them. Now, this is complicated because there's still a concern that if poachers track a rhino and find out that rhino does not have a horn, there's still a chance that they might dispatch that rhino so they don't track that rhino twice. It does not have a horn. However, that doesn't appear to have happened, even though that's kind of a concern for part of this process. Instead, they've actually seen a huge drop in poaching as a result of these programs. However, the Université of Neuchâtel wanted to look at the other implications of removing these horns. So aside from the fact that it's really difficult to do, it puts humans and rhinos at risk. You have to tranquilize them. You have to replace them, or you have to go back and remove them again every 18 to 24 months because, again, they're made out of keratin.
They're growing like nails and hair.
Right. Yeah. So it's not a permanent solution. Oh, I cut off your horn, you're good for life. No. So basically, they wanted to see, are there other impacts on the rhinos from removing their horns? And unfortunately, yes. After loss of a horn, male rhinos reduced their range up to 80% of their territory. So they were essentially more shy. And for females, the reduced range was even greater. So they didn't venture out as far. They also found that black rhinos in general were less social with one another when they did not have a horn. Instead of interacting, they would intentionally avoid one another. So
it's like a teenager with a zit on their forehead. They're
Or
just like, I'm going to stay home until this clears up.
Yep.
It's you're going to a knife fight with a spoon, basically. Like it's
What?
No, they're not even going to the knife fight. They are staying
exactly.
home.
They're saying I'm not going to go to the knife fight because all I have
no.
is a spoon. It's the rhinos. The way they protect themselves from each other as well is with the horn. It's how they intimidate one another. Yes. They size each other up based on the size. So this is also going to impact mating selection. Yes. It's a huge impact. This is what makes a rhino. This is their whole ecology is based on having a horn. So now the question is, do you weigh the gain of poachers not poaching versus changing the species completely? Because you're going to change sexual selection and selective pressures.
Yeah.
And you're going to change the behavior of the species.
Yeah, I mean, and this gets back to the real question of, when it comes down to it, what is a species? And what do we care about when we're talking about genetic diversity? And part of the rhino's behavior is to the males and the females to expand, even though they do have herds, but they expand out so that they can have increased possibility of interacting with other individuals from outside their genetic pool to increase diversity in the overall population, right? Mm-hmm. So you have these territories, you have all this stuff, basic ecology 101. But in this case, I think it's a very interesting question because what you're saying is that the horn removal is actually benefiting conservation from two angles, that they're not being hunted because they have no horn, and they're not leaving their territory. So they're not potentially going to be moving out into an unprotected, unconserved area.
Fair. Yes.
That's
very fair.
But I take your point of behavior and population changes and genetic
bottlenecks. Yes, absolutely. Yeah. So it's, then also you have the question of the whole conversation that we had last week about when you have protected areas, that can impact the behavior in it. Basically, we can't have nice things because we kill them. That's what this is about. If
don't tell the plants in my house.
Yeah, really. If we could just stop killing the animals, that would be great. But there's a huge demand still for rhino horn globally.
Yeah,
why have somebody we not
needs to
past
just address
that?
Like that's the most ridiculous thing.
That would be great. There needs to be a massive influx of funds and resources into specifically working with native populations in those areas, not like
it's cultural identity
rich white people walking in and going, you shouldn't eat rhino horn, right? You need to work with the actual local communities.
Are these local communities that are doing this?
So
it's probably more of the rich parts
so
of
it's
those
a combination
cultures,
of things.
But
There's definitely still local expectations for use of these things. The influx in requests for it is often, yes, because of affluent, often white tourists moving into that space and wanting authentic rhino horn pills or something like that, right? So there still is, yes, it still is, as it often is, the wealthy elite who are paying whatever they want for things that are driving up prices for things. A few isolated communities using some rhino horn a couple thousand years ago didn't drive species to extinction. It's happening now because there's global trade, right? But still, if culturally there was less use of these things and less significance on it, it would be less sought after in the global market.
Yeah.
Yep.
So there needs to be that conversation, and there's been lots of cool research on identifying cultural resources of the animal when they're alive and promoting that and changing. I think we had a whole story about snow leopards about this, right? Changing the the value of that animal. So they're more valuable alive than they are for their resources.
Yeah.
And so that kind of shifts cultural ideals around that animal and can change the way management works. But it's so hard. It's so hard.
And it's definitely not the thing that, you can't have Western elite, quote unquote, scientists coming in and telling locals how to live their lives and what to do. It needs to be from within. It needs to be a community-led conversation, and it needs to be something that changes from the bottom up, not the top down. And I mean, maybe or from both sides, but there needs to be, there needs to be pressure for sure. People are so weird. Changing people's minds is so hard.
Here's how I would just, I kind of agree with everything you just said. However. So my understanding has been that there's a traditional Chinese medicine marketplace for rhino horn.
Mm-hmm.
This is, so then if you've got what you're calling Western white people, coming in and saying, hey, we're going to throw resources at protecting this rhino, you're talking about maybe an argument between two
it depends. There are Asian
very
rhinos.
Wealthy nations.
There are Asian rhinos. So that's part of why this happens, is that there is an expectation that that rhino horn, it does come from there. Is my point. So then there's a global marketplace, but it's based in this interaction that's thousands of years old because there are rhinos in Asia.
Mm-hmm.
In China?
In Asia.
I didn't know.
In Asia.
That's okay.
Yes.
Fine.
But it's much closer than saying that Chinese markets are buying from Africa. That's not how this started.
Okay.
So that's all I'm saying. It's more complicated than that.
And this is one of those places too where, like, you would think the upside of having an authoritarian government would be you could just outlaw a thing with repercussions and it would happen.
Yeah, but not so much because it's still cultural. And so you're dealing with cultural beliefs. And like Blair said, there are people who move into a place or even who are from elsewhere who go, ooh, quote unquote, ancient Asian medicine, ancient Chinese medicine, ancient Indonesian medicine, ancient African medicine, whatever.
It'll cure cancer, or
it'll cure this
help
or whatever.
Me in bed, or whatever, even though it is just
all
keratin.
The money. Yes, and I will give you all my money, and so then there's a market, and there's all beyond just the the specifically cultural market. There it goes beyond that as well, and there's money involved, and so where there's money, there is market, but it's cultural overall, and so you have to get past those cultural beliefs that you're taking away something from people that would help them.
Regardless, we're not going to solve this tonight, but what we have found out is that some of the current mitigation methods that just try to put barriers in between poachers and rhinos are more complicated than previously thought.
Yeah, I gotta think that through. Complications. Well, okay, speaking of like evolutionary complications, where the heck did eukaryotes come from? Wait.
A one cell ate another cell.
Yeah, okay, so it's like some sort of symbiosis. So you have the prokaryotes, which are the one-celled organisms. Suddenly, in history, they're like, we're gonna be multicellular! We need more of us in us. And that happened, and now we're here. But how did we get here? We don't really know. And so researchers are trying to trace our evolutionary history down to that root, that microbial root of the linkage between the single-celled organism and our multicellular ancestors. Again, published in Nature. This is, you'd think I just read Nature, but again, published in Nature this week. Researchers at the University of Texas, Austin, in collaboration with a bunch of other universities, among which are the Wageningen University in the Netherlands.
Yeah, that one.
That one. My peoples. Anyway, the researchers have been delving into the microbial world of archaea bacteria, these single-celled organisms that aren't just microbes, they are extremophiles. They are known to have evolved in the hot vents at the bottom of the sea. They're known to be evolving around volcanoes, in acidic pools. There has been a lot of work in trying to figure out what's up with the archaea, because they're like their own special cool group of bacteria. And so the researchers, many researchers, have been for years dredging the bottom of the oceans, taking samples and trying to get genetic samples, archaea samples, to be able to bring back to the lab and grow in the lab. And to date, only 2 species of archaea have been able to, they've only been able to grow 2 species of archaea in the lab. So that limits the way that researchers are able to understand how archaea link to everything else. And so this new study, researchers, instead of actually growing them in the lab, they did one of those genomic samples where they took a bunch of samples from a lot of places under the ocean and got over 50 undescribed genomes. And specifically, these archaea genomes are of what are called the Asgardians. They are a specific...
What is that?
Yeah. So,
Thorn Company.
So once upon a time, researchers decided that they were going to have fun with naming extremophiles. And they're like, wouldn't it be really cool if we named them after Norse gods?
Yay!
So there's an entire family of these Archaeans, which are Asgardians. And they've been describing these Asgardians that they've been finding in volcanic vents and hot smokers and other things from the bottom of the ocean for a while now, but they haven't really found that evidence that connects not the Asgardians, but the archaea generally to the rest of eukaryotic life. Although it seems like there's been this hint there. And so a bunch of researchers thought there were a group called of the Loki Asgardians that may have had molecular and cellular mechanisms that could have led to eukaryotic mechanisms. But this particular study, they were able to get over 50 different genetic signatures from their genetic samples. And what they have determined is that, nope, Loki was not the lead god in determining eukaryotes. In fact, they have been able to isolate the probable initiation of eukaryotes, multicellular organisms, from what they call the Heimdall Asgardians. So they think that the last common ancestor of Asgard archaea was probably thermophilic chemolithotroph, and that the lineage from which eukaryotes evolved adapted to mesophilic conditions and acquired the genetic potential to support a heterotrophic lifestyle. And yeah, so in their analysis, they have determined that Hodor, not from Game of Thrones, but the Hodarchiates, Hodarchiales are the group from which eukarya expanded because they share a number of ribosomal proteins in common, and the mutations of those seem to have a lot of evolutionary lineage. So bottom line, there's a group of extremophile bacteria, single cellular, live at the bottom of the ocean, crazy environments, archaebacteria. One group of those are named after Norse gods. They are the Asgardians. And one of those, the Hodarkales, or however you say it, the Hodarkales are the sister group of eukaryotes. And that eukaryotes, which are multicellular, which we are, that's where we came from.
From extremophiles. That makes sense.
We come from extremophiles. I love this. Yes.
Our planet was extreme.
Yeah. Extreme. And this may explain extreme sport popularity or not. I don't know. But I just love that people have been following this line of inquiry for a while, that at one point in time we didn't know about this group of prokaryotes. And then they showed up on the scene and everyone went, "Woo, what are these? Oh, we can't really get them to work in the lab. We can't grow them. They are extreme. We only find them in these crazy places. How do they relate to the rest of everything?" And for a while it was like, they are a third group of life. That it's not just prokaryotes and eukaryotes. It's prokaryotes, archaea, and eukaryotes. But now we're saying that they were the stepping stone between. So,
Interesting.
Yeah.
Nice.
I still think it bothers me that we would, anytime that we're looking at things that are current, it's tough because we have the based on morphology, based on how something's put together, this came from that. But we're all evolved at the same length of time from the beginning, so there's
changes that have taken place in everything.
Yeah, there's changes that take place all over the place. So you look for those signatures that suggest relatedness.
Yes.
And that's but it's
until you get that time machine.
Yeah. So the thing with the HODs, which the scientists are calling them for short, is that they instead of digesting methane and living in darkness, they actually have a different metabolic pathway that is more in line with the way that the majority of eukaryotes metabolize energy. And so that is what led, even though this whole group of archaea evolved like 2 billion years ago, further back than 2 billion years ago, it is this similarity in metabolism, which is something that is very, very, I don't know, it's very specific and important to the survival of an organism, right? So how do you live? How do you use energy? What do you use? Carbon dioxide? Do you use oxygen? Do you use what is it that you use? And everything falls from there.
The problem I have with it is it's 2 billion years of evolution of a non-multicellular life form that could have the convergent evolution aspects do not need to be broad.
But from what we've talked about in the past, we also know about molecular clocks. And we also know that there are specific genes and there are ways that mutations happen within genes that can be timed and so we have baseline rates for how mutations occur in different species. And there are lots of ways that researchers are looking at this to be able to come to this determination that isn't necessarily clear as we're just looking at it on the face of it, but when you understand the methodology. Yeah. And I know you've been looking and you've taken classes in this stuff. So,
well, I don't know anything about, what do you call it? Extremo
archaeology
or whatever you're talking about there. I don't know if they do horizontal gene transfer for example. Like I don't.
All of that stuff is probably still going to do that kind of thing.
If
so,
that's still there, like that's why
right.
even looking at bacteria and trying to say like what bacteria begat what bacteria, like you can kind of create these big groupings, but gosh, they can as soon as something can just swap a segment of DNA with something else, your whole biological molecular clock thing, your whole phenology, like it makes it very difficult to really say anything for certain going back a billion years.
I think it's fair enough to say that. And I mean,
we do our best. We don't have a time machine, so you gotta at a certain point, if you want to try to figure out what happened, you have to make some guesses.
This is
absolutely.
This is the morphology of the modern age.
Yeah, and I know that there are also a lot of versions of theory on this. That's why it's like
a lot of phylogenetic evolutionary conclusions that were drawn with animals were wrong. A lot of them were right.
Yeah.
So it's you gotta you have to start somewhere.
Of course. I'm just throwing in a little head scratch, not because I'm trying to say that this is in any way not the correct or best theory that is possible.
But there's always questions.
I just have a lot of questions because I don't understand how you can say anything definitive about that long ago.
I think it looks like and we hypothesize that is implied in this case.
Of course.
Yes.
Whenever you add some billions of years, you're like, well.
Speaking of billions of years, well, not billions of years. How about hundreds of thousands of years? Tell me about Lucy, Justin.
3.2 million-ish years ago. Yeah,
really?
We can
that long ago?
Millions.
Lucy's old.
Millions of years ago. Well, she was found in the 70s, though. Which, if you were born in the 70s, then you might feel like it's been millions of years.
Yeah. Mm-hmm.
So this is a Cambridge University researcher. She has digitally reconstructed the soft tissue of Lucy, the Australopithecus afarensis, who was actually named after the Beatles song "Lucy in the Sky with Diamonds," after she was discovered in Ethiopia in the mid-70s. Lucy is one of the most complete examples of an Australopithecus skeleton. They have recovered about 40% of the bones in that find. This is an early species of human hominin that lived in East Africa over 3 million years ago, right? So, shorter than us, ape-like face and smaller brain, but able to walk on 2 legs. And real quick, this is, I just wanted to point out, ape-like face that is in the press release. I don't believe it's in the paper, but is in the press release of the story. This is a nonsense term, I think, because I just want to point out that there are around 20 species of apes on the planet.
And they all look
including
different?
Humans?
Yeah, and with global population of apes is about 8.5 billion, with 8 billion of them being humans.
Yeah, exactly.
So, ape-like mostly means human. Like, current modern human. That is the most...
That's definitely one of those terms where they put humans to the side as not an animal.
Yeah.
Humans are over here, animals are over here. So it's, yes, I find that very common that people forget that we are.
I think Lucy is a dead ringer for Christy Brinkley. Come on.
And they also almost always, when they say ape-like, mean chimpanzee.
Yes.
Like, almost always. And there's less than, there's, again, another 19 species of apes that have different morphologies. Anyway, press release goes on. Some have argued that she moved around in a crouching waddle, similar to chimpanzees, our common ancestor, when they walked on 2 legs. So I pull the record up again. So, this is the press release. Whoever's writing the press release at Cambridge University. This is not the paper. There is no chimpanzee ancestor. of humans, nor of Australopithecus. Chimpanzees
And
are as
humans
evolved as current modern
share
humans.
a common ancestor.
We
Yeah.
share a common ancestor who chimpanzees are probably a lot less like than current modern humans in that the whole knuckle walking aspect of chimpanzees, gorillas, and some extinct apes is something they evolved. And the upright walking without knuckle dragging likely came from having been arboreal and then being bipedal on the ground as well. Being bipedal in trees before being bipedal on the ground.
Okay. So, but this story is about how Lucy stood, right? And so that's, I feel like usually they always talk about where the spine attaches to the skull when it comes to this sort of thing. Is that what this was about?
Nope.
No, they went the other way. This is working focused on the legs. So what they did is they reconstructed the legs, the leg muscles and the hip muscles, and the researcher kind of put those in. But I just wanted to point out the chimpanzee thing. That is the cartoon of evolution that everybody needs to get rid of in their head. That is not the thing.
And I'm going to say that looking at some of these figures from the study in the Royal Society's open access journal, it looks like people legs. Like, they're models. This is like, I'm like, okay, how different is that from anyone else?
Right, and so there is a little bit of uncertainty in this because you're placing a whole bunch of muscles in places without having any of the soft tissue as an example. So the reconstruction,
but you know where they connect because you have all those bumps and divots where the ligaments and tendons, like where everything would connect to the bones.
To an extent. So they did use a human form basis for reconstructing the musculature. If a chimpanzee form had been selected as the basis of reconstruction, it might have been a little bit different because it would have been using chimpanzee muscles and trying to get them to fit in, as an example. However, it would have there are more similarities with human than there are so there's more dissimilarities with the chimpanzee. And it would have pushed it to a more human musculature in the end anyway. Because one of the very conserved aspects is the way that the vertebrae muscles, the spinal muscles surrounding the vertebrae are going to connect. And so how these connect, along with, I think, 36 muscles in each leg, what it did is it basically ended up creating a reconstruction that showed that Lucy could stand straight, erect, and lock knees just like a current modern human. Lucy's knee extensor muscles and the leverage they would allow confirm the ability to straighten knee joints just as much as a current modern human. And this has actually been adding up to, there's been other side research and other ancient ape finds and the like that has been pointing to the direction of bipedalism is not being something that we developed once we decided to start exploring beyond trees, but it's something that developed in the trees, in an arboreal, or a combination of arboreal. Anyway, but this is the first time that this has been shown in Lucy. That Lucy,
I'm jealous of Lucy's gluteus.
Lucy had Lucy's not. Yeah. Lucy's got bigger muscles, bigger leg muscles in a lot of ways. And was also, if you look into the upper body, it would have been more able to climb in trees. Stronger upper body, or however, the way the joints are set in there. But yeah.
So back to my previous kind of comment about how they've always talked about whether a specific hominid or relative walked upright or not, they always looked at the skull and the spine, right? So
it's part of connection. point.
So, did she just have a crazy hump? Did she walk like an old man with a walker? Like, did her spine still connect to the back of her head? Because I thought that's why they were sure that she wasn't bipedal.
She had long arms still, I think. But yeah, I think, and because she had such a large pelvis, they expected it to be angled back in a way that would have put her at a quadrupedal predominance.
It feels like we have some conflicting information. So, like, was she in between? Did she do a little both? And also all of the fossils that we've looked at, where we've only looked at skull spine connection, location, and angle.
Mm-hmm.
Does that mean now that you have to look also at the legs? Sounds like yes.
Yeah, and part of the problem is, of course, having the complete fossils, which this is the most complete, and it's 40%, and it's But
yeah,
so this was actually all this research that we're looking at here is generated from, they took all of the scannings and 3D renderings of Lucy and put it online in an open access resource.
That's
and so
cool.
This Cambridge University researcher basically downloaded it and started applying the muscles in the right locations with expertise.
And I think the big assumption historically is that humans are related to chimpanzees, like you brought up in the very beginning there, which we need to get out of our heads. And
we're related. We're still related,
but they're our ancestor. And if you start from the assumption that chimpanzees are our ancestor, you are incorrectly attributing certain body morphology. You're incorrectly attributing behaviors and all sorts of stuff. And so, for, decades, we've probably been looking at Lucy all wrong.
For 50 years.
And she's like, what? I was alive for like 3 billion. Come on. I mean, million.
A gazillion.
A gazillion. She's an old lady. She's like, she doesn't have to put up with this stuff anymore. She's like, I don't care. I have purple hair. Just be quiet. You kids these days.
Well, like all of our misnomers, that long ago we would have been, not walking upright. Well, that's not a thing. It turns out, hominids walked upright. That's just a thing. Hominids. Oh, small brains couldn't do this, that, or the other. Small brains can do everything that current modern humans can do, and in fact, what Blair's been explaining is doesn't even need to be a human brain to do a lot of human type activities, but we attribute to intelligence. So.
Yeah.
We're having to reinterpret all the time.
All the time. And now for kangaroos as well. Published in Alcaringa, an Australasian journal of paleontology, researchers have reviewed the literature on kangaroos. Living kangaroos and extinct kangaroos.
Macropods.
Yes, the ancient fossil evidence of kangaroos to determine, did kangaroos always hop? And the answer is no. Hopping was not necessarily an initial kangaroo trait. And in fact, according to their analysis, it is something that really ancient kangaroos didn't do, because ancient kangaroos were generally bigger than modern day kangaroos.
Kangaroos are huge!
Yeah, there were some real big ones.
There were some old
big
ones.
Carnivorous.
Modern kangaroos are still big, under 100 kilograms, but they're not as big as some of the ancient ones. But even the littler, like modern day kangaroos, it's from the wallabies to the kangaroos. Everyone's like, I'm gonna hop, hop, hop all over the place. But apparently, according to their review, this is just something that came with the smaller sizes. That the evolution of using the tail as a fifth limb and also the smaller sizing changed the way that the ankle bone, the calcaneal heel, was developed to support weight.
Aha.
So,
Okay, so this is interesting.
Yes.
So, my understanding of why kangaroos cannot walk and must hop is because they do not have a kneecap. Their
which is
two leg
possible.
bones overlap, and so they can't extend out enough to straighten their leg to walk. So this is really interesting. This isn't even looking at the kneecap. This is looking at the ankle.
This is looking at the ankle, not even the kneecap. Yeah, but I think that's an interesting point also. But historically, the ancient species didn't have that lever arm counterbalance, so their tail, and they didn't have that balance, and the ankles were not set up to deal with those forces. They had a thick Achilles tendon, and so that wasn't really good for the elasticity that's necessary for the hop, the jump, the jump, the jump, the jump. So it's a much stronger tendon, but not for hopping. And so, I mean, it's really interesting because now I'm envisioning these big, fanged, giant fanged kangaroos lumbering at me with a bipedal step as opposed to hopping, and it's a little more terrifying. But, but yeah, old big kangaroos were striders, not hoppers. Their ankles didn't support it.
Mm-hmm. Very interesting.
Yeah,
yeah. I want to see the fossil knees. Pan up. I want to see. Call these researchers.
You gotta look at that. Come on. I'm sure it's part of it too, but they only looked at the calcaneal heel and its ability to support forces and weight that would be involved in hopping compared to modern day species. Yeah. And then finally, have you been to a bar that the bartender goes, ha ha ha, I'm going to dump some peanuts in your beer when you order it. And then the peanuts are like, no, I sink to the bottom and then I rise to the top. And then I sink to the bottom and I rise to the top. And the peanuts are like doing this magic peanut dance in your beer. No?
No.
Okay. Roasted shelled peanuts. Put them in a beer. They do a little dance. It's the beer peanut
in the
dance.
shell? Or
No.
Shelled
out of the shell.
Take the little peanut.
The peanut meat.
Out of the shell.
Take the little peanut, throw it in the glass. Peanut's gonna do a little peanut dance. Woo!
And it'll do what now?
A little peanut dance.
Oh, good, there's video here.
I got a video for you. You don't just for
like
up and down. And I would assume that has to do with the oil content.
Nothing to do with the oil content.
Okay.
And so that is this story in a nutshell. Researchers decided they wanted to explain why peanuts do their little up and down dance. And it was a Brazilian researcher, Luiz Pereira. And so he he had the idea when he was first passing through Argentina's capital, Buenos Aires, to learn Spanish. He said it was a bartender thing in the city to take peanuts, pop them into beers, and the beers do this thing. And he was like, what is going on? So they have looked into the peanut dance, and he's now working at Germany's Ludwig Maximilian University of Munich.
Is it the bubbles, the carbonated bubbles that are rising him? And then they pop and then they fall?
Got it. Yes, nucleation. And so the peanuts, maybe because of their oil content, are more likely to get the little bubbles, the nucleation sites within the carbonated beverage than the sides of the glass. So the bubbles all form on the peanuts, and it depends on the angle of the bubble as it forms on the peanut and how it's able to loft the peanut up to the surface where the carbon dioxide is released, and then the peanut can sink back down to form more nucleation sites and bubbles, and so it goes up and down within the beer.
So would this work on a jelly bean?
Possibly. Yeah. We should try it. We could do peanut.
It's about the shape is basically the idea, right?
The shape, but the weight, I think, is also got to be right.
They call it, in their paper in the Royal Science Open Science Journal, their beer gas peanut system. And so they have not looked at jelly beans.
Well,
We
that's what I want to know. What makes the
We know-
peanut special, and what else can I do this with?
We
I
should
have
try
a feeling.
all the things.
I have a feeling a jelly bean is too dense and
Yeah,
would
probably.
sink.
Yeah.
But yeah, now
And then, like, what else would be equivalent?
Right, what else can we put into the beer? I really don't want a jelly bean in my beer, though.
I think a more important question is: do
How
we
about a
need
banana
more
flavored
than
jelly
just the
bean?
peanut to be able to do this trick? But
Yeah, Kiki, what's the practicality of this research? Did they talk about like what they could apply this to for war or exploration? Because it's always one of those, right?
It's not war. It has to do with mining the earth for its resources. So the idea is
option number 3. Got it.
Yeah, that if right now we use air injected into a mixture where there's a mineral like iron, and the air will attach to it and other minerals sink to the bottom, so it can allow separation of different resources in a resource not manufacturing, but isolation situation.
So you got to carbonate the water.
Got to carbonate the water. That's
Debbie's got it in the YouTube chat room. A coffee bean. Maybe that would work.
Oh, I bet a
that would
coffee
also be tastier than putting peanuts in beer, I think. Especially if you had like a nice stout.
No. You're missing it, Blair. Tastier than a jelly bean in your beer.
Okay.
Yeah.
Sure. I want to get the peanuts out of there. No.
No nuts in your beer. Coffee bean. That'll be great.
I could do that. I could definitely do a coffee bean in there. Yeah.
See if it works. It's oily. It's still a bean.
Yeah.
Has lots of like similar weight issues. Also very similarly curved. Coffee beans have an interesting little cut down the middle that could also provide some interesting dynamics.
So in this mineral extraction zone, we're going to carbonate the water and skim the minerals off the top foam of this bubbling water with like a giant like net.
Imagine magma. It's not beer, it's magma.
Okay.
And we are learning how to
carbonate the magma.
We're carbonating the magma. We're learning how to get...
no.
We're going to get what we need out of it. But in the meantime, I highly recommend let's figure out what else does the peanut dance in your beer.
This is my other question. Does it have to be beer?
No, this is just something from a bartending thing. So it could be anything carbonate. We know Mentos are incredible sources of nucleation. And when you put them into Diet Coke, suddenly you have this massive, explosion almost of the carbonated gases that come out. So this is just a different take on that. Different beverage, different carbonation levels, different source of nucleation sites. But understanding the dynamics of how putting a substance that leads to nucleation into another substance that has its own viscosity and its own traits, then suddenly we can start to look at things that could be used for, mining minerals or doing things in a more efficient manner. Hopefully, right?
Interesting.
Yeah.
Okay.
Yeah.
Sure. Just an excuse for researchers to drink beer and eat peanuts as part of their experiment. But you know what? You guys have fun.
Right there. Yes. It'll be a great time. This is This Week in Science. Thank you all for joining us for this first bit of the show. We have a few more stories to go. If you are enjoying the show, please head over to TWIS.org and click on the Patreon link. You can choose your level of support, and we wonderfully thank you for helping us maintain the show and keep going week after week, year after year. It's you who really help us get to where we are, and we do this show for you. We love talking to each other, but we love seeing you in the chat rooms. We love getting your emails. We love that we get to be creating the show because you're here. Thank you for your support. We can't do this without you. Oh, and by the way, if you go to the Zazzle link, you can get a mug like this one that I'm drinking coffee out of at 9:30 at night. I'm not sleeping. Am I? Nope. Nope, not gonna sleep
That's
tonight.
not coffee. I don't believe you.
It's coffee.
Okay.
I swear it's coffee.
Definitely your funeral.
All right, coming on back. This Week in Science. It's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. Animal Corner.
What you got, Blair?
Oh my goodness. Well, I'm going to start with a fruit fly funeral, actually.
Oh.
Not a funeral, just,
Taking it down
a
a
fruit
notch.
fly that sees another fruit fly die, which I imagine happens quite often.
I just try to kill them in my kitchen, but really, yeah.
This is from University of Michigan. They wanted to look at the link between death perception and reduced aging in flies. Not depth perception, death perception, which is something that is studied. It is how animals respond, both in behavior, but also internally, how their body physically responds to witnessing death. And yes, this is a thing that is studied.
Well, we've, like, crows have, corvids have funerals, elephants have funerals.
Right, so there's the behavioral side, which is, do they recognize the death and do they act a certain way about it? This study is actually about what happens in the fruit fly's body as a result of witnessing death. They found in previous experiments that perceptual experiences can affect aging. How that happens is still a mystery. But so previously, University of Michigan reported that when fruit flies see other dead fruit flies, they experience advanced aging. And they even were able to drill down and see that this effect depends on a type of serotonin receptor. So there's something happening in their brain as a result of seeing death that makes them age faster.
No, why? Wouldn't it be like, oh, there's death, I want to live? Like, why?
So if you're looking for that, I don't have it for you. I really hope science figures this out soon, because I did this. I spent way too long trying to figure out what other species links there are between death perception and aging, and I couldn't find anything. Seems like historically they've really only seen this in flies so far, but this is something that they want to kind of drill down to the mechanics of and then see what's happening in other species as well. But so what's happening is they found out in this new study a specific group of brain cells in the fly called R2 and R4 neurons are activated when they encounter other dead flies. And that specific increased activity leads to rapid aging. They figured this out through fluorescent tagging. And what's even more specific to this is that they silenced ring neurons in the region to see that R2 and R4 were having this impact, and when they were artificially activated, lifespans decreased, even when they didn't see death. So R2 and R4 are causing this as a result of seeing death. They're confident about that because they took the death out of the equation, just turned these guys on. They age faster.
Straight to the molecular signaling, and it still happens.
Yeah, exactly. So why? I don't know. Evolutionarily, why would this happen? I don't know.
Is it olfactory? Is it only vision? Like, what is it that's making these neurons go?
We need to find out.
Yeah.
But even more specifically, the researchers think that because they found out what's going on in these brains, they could reverse engineer it to turn them off to lead to targeted drug therapies, potentially even in humans, to slow aging.
What?
So if you figure out what's speeding it up, reverse engineer that, turn it off,
No, wait, what?
Can you slow down aging?
So,
But do they know these things affect aging in humans?
No.
No.
So that's
No, but-
20
but if-
studies from now, but still.
It is, but still, like, if we were to extrapolate
Yeah.
this and
Yes,
say,
exactly. That's
oh,
what
it
we do
applies
here.
to humans, what it would then mean is that we need a lot less violence on television.
Well, so that's
Right?
one of the things, one of the very first things I thought of course, because he lives in my house, is my significant other sees death a fair amount.
Oh,
Is
no.
that
as a
impacting his
paramedic.
aging?
But what about you going to a graveyard or visiting your grandparents?
If you're seeing dead
Or
people at a graveyard, that
watching
graveyard has been
movie.
constructed very poorly.
Well, how about open caskets? There's all of those things, right? Like, I think it's very...
We're not fruit flies. That's all
We're
I'm
not-
going to say.
We're not fruit flies. We can reason through things very differently, right? So it's a lot more than just, oh, there's something dead over there, right? So there's more going on in our brains. I think there is less expectation from these researchers that they could extrapolate the impact of death perception to humans, and more expectation that they could extrapolate what's going on in the brain related to aging to humans. However, I still want both. I want more research on death perception in other animals, specifically leading up to birds and mammals. I want to figure out what's going on there. Because also there's a fascination with death, like you were talking about, Kiki, like crows and elephants, both are very fixated on it when it happens and perform rituals related to it and all these things. So is that being counterproductive because there's an impact from seeing death? Like calling more friends over to see that death, is that actually observing that death hurting the That can't be true, because then evolutionarily they wouldn't do that anymore.
Yeah, so the idea with crows is that they have their crow funerals because what's happening is they are identifying a threat to other crows.
Mm-hmm.
Look here, a crow died. Be aware,
Right.
and don't die also. Yes,
Yes.
It's basically
live and
like
learn. Yes.
Yeah, exactly.
So yes, I was expecting the opposite from this study when I read the headline. I was expecting that aging decreased when they saw dead flies because of that, learning from what happened, but also exactly what you're talking about. Like, oh, hey, less competition, time to really live it up.
But maybe that's it also, is that they saw death, they saw other dead flies, and those neurons got stimulated, and they were neurons that didn't just lead to death, but increased their metabolism so that they worked harder to reproduce faster, and that wasn't
interesting.
something that the researchers measured.
Yes, I love that. Okay, that needs to be looked at.
Yeah,
Yeah.
Kiki, that's a great insight.
That it wasn't just the death, it's the metabolism gets sped up, and it's like, you need to go do the life thing now.
Yes, there's an urgency. Death is on. It's a midlife crisis. You see
Death is
death,
an
and you're like, you gotta
aphrodisiac.
take care of it. Yeah. Wow, interesting. I love that.
I don't know.
Well, I'm gonna
Yeah,
be
I think-
keeping my eyes peeled for more death perception in science news in coming years.
Bring us your depth perception of the death perception.
There you
Yes.
go. Last for the animal corner, I wanted to talk about sand. It's coarse, it gets everywhere. And in the case of sand and microplastics, they get hot. This is from Florida State University, and they wanted to look at what concentrations of microplastics do to the temperature of sand. This is something that hasn't really been looked at before, and how the presence of microplastics affect the thermal profile of sand. Why do we care, aside from just hurting our feet when we go to the beach? Well, let's talk about sea turtles. Sea turtles, they lay their eggs in the sand. And as we've discussed many times on this show, the sex of the babies are not dependent on genetics, like in humans, an X or a Y from dad. You always get an X from mom, but an X or a Y from dad, right? Instead, it is the temperature. cooler babies are male, warmer babies are female. And so usually pre-climate change, you'd have this nice gradient in your nest where you'd get about 50-50 coming out of the nest in the end, because there's certain areas closer to the top that are warmer, lower down they're cooler, towards the center they're warmer, towards the outside they're cooler, and it kind of creates this really nice gradient. So because of climate change, seeing an increase in female turtles, that causes a problem with sex ratios, harder to find a male, harder to have babies, impacts the general population. But what they found by mixing sand from beaches in Florida and then mixing it with black or white microplastics is that the microplastics themselves also impact the temperature of the sand. So concentrations range from 5% to 30%. They recorded samples or temperatures July through September outside by burying digital thermometers at the same depth at which loggerhead sea turtles typically lay eggs. Samples with higher microplastic concentrations had greater increases in temperature, so those plastics are really holding on to the heat. Samples containing 30% specifically of the black microplastic pieces had the highest mean difference. That totally makes sense. Those samples were 0.58 degrees Celsius warmer. That is pretty substantial. And
fascinating to me. I mean, I know that, I mean,
that
we have
is
like
a lot.
There are like the glass sand beaches where it's obvious that like glass has been thrown out of ships and other places and been ground down to sand. And we know that sand itself is silica, and but the fact that the plastic is holding on to heat that much more.
Yes. And so it's 30% though. The kind of asterisk here, that's a crazy concentration of microplastics. 30%, a third, right? So for every 3 sand particles, there's 1 microplastic. That's crazy. That's about 9.8 million pieces per cubic meter.
What's the normal that's found on beaches?
Yes. So current research found the highest reported concentrations collected from beaches is about 1.8 million pieces per cubic meter. So about what is that? An eighth of that, right?
Yeah.
So it's not. No, that's not an eighth. That's a fourth. Math. It's closer to a fifth. Anyway, it's about a fifth. But there hasn't been a lot of research on microplastic concentrations in sand
at all.
So we don't know what it actually is worldwide. Also, it's going up. So recognizing that you can get there still helps to kind of recognize the urgency of this issue. And so, keeping in mind, nesting grounds are incubating eggs are around 29 degrees Celsius, below male, above female. So smaller concentrations of plastic, it might not create a half a degree Celsius change, but it would still create a change. And that change could be enough, especially in combination with climate change, to push the sex ratio even further in the wrong direction. So it's, not great news.
No. I mean, if you've ever tried to walk across a hot sand beach in the afternoon barefoot, you know what kind of temperatures you're dealing with. And if not people, if sea turtles and birds and others are burying their young in that sand, then that's going to have major impacts, or they're going to move other places if they can in time.
And keep in mind, this is just another way that plastics are messing with animals. We talk all the time about ingesting, bioaccumulation, increased cancer risk, all these sorts of things. Changing the temperature of the environment. That's just something else we haven't really thought about.
I'm going to keep that tagline that you used there. Not great news.
Not great.
It's a wonderful way to start a lot of these stories.
Not great. Not great.
Yeah, but one good side of it is that if it's that hot, then bacteria probably can't live in the sand, so there's fewer bacterial infections possible. But anyway.
Might also impact the microbiome of turtles coming out of the shell, though. So that's I don't know about that.
Yeah, well, and I would just
so many
add,
things we don't know.
If you're talking about the surface, having that increased temperature? Maybe. If you're talking about a little bit further down where temperatures normally would be cooler, it might actually increase microbial activity if it's getting them into a better temperature range, more comfortable temperature. So it's
interesting. Yeah, really interesting point.
Generally, though, the plastic in the beach, which is the small particles, not good. Can we stop putting the plastics in the ocean?
Less plastic would be great.
That'd be great.
Yeah.
Thank you for the Animal Corner, Blair.
You're
Justin,
welcome.
where are you going to take us now?
I'm going to take us directly to not great news. Here's something here's something I didn't know to consider, wish I hadn't learned about.
And
it actually fits very much with what the story you were talking about there, Blair. It turns out about 25% of global carbon emissions are captured by forests, grasslands, and vegetation in general, which is good news. During photosynthesis, plants store carbon in their cell walls as well as in the soil. Because of this, soils tend to contain twice as much carbon as the atmosphere does. Deeper subsoils, more than 8 inches, account for roughly half of the soil carbon, which means there's quite a lot there, but there's also quite a bit right on top. Scientists at the Lawrence Berkeley National Laboratory, Berkeley Lab, and University of Zurich have revealed that the organic compounds that are present in carbon sequestration in deep soil are more vulnerable to decomposition during global warming. This is Margaret Torn, a senior scientist at Berkeley Lab, Earth Environmental Sciences area, and the senior author of the study. Our study shows that climate change will affect all aspects of soil carbon and nutrient cycling. It also shows that in terms of carbon sequestration, there's no silver bullet. If we want soil to sustain carbon sequestration in a warming world, we will need better soil management practices, which means minimal disturbances of soil during forest management and agriculture. So basically what they did is they went to the Sierras and this research forest. It's the California's Brigade Forest Research Station in the foothills of the Sierras. And they stuck 1 meter deep plots of soil with these heating rods that increased temperatures by 4 degrees Celsius. Very reminiscent of the story we just heard. And that is also the realistic amount of warming projected by the end of the 21st century due to greenhouse gas emissions causing global warming. They found that 4.5 years of warming at this temperature led to large changes in carbon stock at depths down to even 30 centimeters or 12 inches below the soil surface. Basically, it was a 17% reduction in lignin, which is a compound that plants use to form rigidity.
Yeah, important for strong trees.
Mm-hmm.
Yes, very important.
Nearly 30% loss in cutin and suberin, the waxy compounds on leaves and stems and roots that protect the plants from pathogens, as well as significant losses of pyrogenic carbon. So it had been assumed that pyrogenic carbon, this is the carbon that's like after a wildfire and then it kind of goes down to the soil, burnt stuff. There was an idea that this was a very safe, stable form of sequestered carbon. But they found that in the deep soils that were heated, they were broken down, and that carbon was released. So, it's with this thing. We've been thinking like, the permafrost, and there's methane in the ice, and that's going to get released, and maybe there's carbon underneath the ice in places that's going to get released. But the heating of just soil everywhere, and knowing that like there's half of the atmospheric carbon is sitting in that top layer of soil. Oh, that means things could be even worse than the bad news. So not great news.
Not really not great news at all. But
one of the things that could counter this, good cover. Yes. So if you've got trees covering the soil, it won't heat up as much.
Yeah, I was just going to say that. I was going to say that if the trees are there, they're providing shade, and that shade is going to keep the soil below cooler, and it's a complete difference from open areas.
And my final story of the day, turns out smoking has been killing non-smokers.
Who knew?
People who've never smoked.
You just talking about secondhand smoke or what?
No, gosh, no. That's a reason to smoke. Secondhand smoke is so bad. You want the first-hand smoke because apparently that's better.
Oh my!
No, this is published in JAMA Network Open. Researchers have looked into mortality estimations, mortality charts that you can go and look up. To tell you. If you're this age, your chances of dying and that sort of thing. Turns out this is one that's on the National Cancer Institute's website called Know Your Chances. They don't take into account smoking status in any of the data or any of the estimates. Nope.
Really?
It's not that they're leaving smokers out. Smokers are included, but they're doing a general population analysis. So it's all people. Now, it shouldn't be a big deal. Smokers make up, 11.5% of the population in the United States now, down from over 50% in the 1940s. So tremendous progress there.
Wow.
However, what they found was that 11.5% of smokers that are out there are weighting mortality estimates considerably for everyone else involved, especially with smoking-related diseases.
So they're pulling down the medians, is what you're saying.
So this is not even just doing the analysis I'm about to talk about here isn't just on smoking-related diseases. This is all death average risk of all death over 10 years of any cause. The authors give an example of a 60-year-old white male with the general population average death risk of any cause over 10 years being 14.5%, which is kind of high if you just looked at that number because, the considered death rate once you hit like 70 years old or something is somewhere around 8% a year or something in that ballpark. So 14.5 seems kind of high. It says that if an individual were a lifelong non-smoker, adjusted risk, getting the smokers out of there and the former smokers, the adjusted risk goes down from 14.5% to 9.7%. That's significant. Yeah. A former smoker, somebody who's quit smoking for more than 2 years, goes down still to 13.2%, which means those folks who are smoking are weighting everybody else up. So what is it if you were just a smoker? 60-year-old white male smoker, it's a 10-year risk of death, by the way. The 70-year-olds is a yearly. The risk jumps to 27.3% over 10 years. Nearly twice the general population average, almost 3 times the risk for a never smoker. So, anyway. Also, if you take the smokers out of the equation, this is okay. So for lifelong non-smoking women, the 10-year risk of breast cancer was slightly above all other causes. For women who smoke, lung cancer and coronary heart disease took the lead as the causing. So breast cancer isn't even their most high risk category at 30 to 35. Anyway. Avoiding mortality is hard enough as it is.
You can't avoid mortality. It's going to happen.
Well, you can. Actually, everybody, both of you and everybody listening right now to this show has avoided mortality every day of their life.
Yes, but I mean, eventually the mortality is coming. It is birth, death, mortality is coming. at this point, you can look at your days and go, "Yay, I survived today."
Yeah, but the thing that I
amazing."
think this study is pointing out is that it's not the thing at the end that gets you.
But it is.
It isn't.
It's the other people.
It's all the things that came before that add up.
Yeah.
To
Makes
that
you
end.
more likely to get got. It's what they call comorbidities, right?
Anyway.
So
that's why if you
Risk
look at the mortality
factors.
statistics, everybody should most people, 88.5% of people should be doing better than the estimates. It's just that 11.5% that's bringing it down for everyone.
Just, don't drink, don't smoke. What you gonna do? Don't drink, don't
eat
smoke.
healthy.
Yeah.
Don't do risky things.
Just stay. Bed rot. We should all be bed rotting.
No, that's bad for you also.
No, it's not. You want to bed rot. Just do the bed rot. That's fine. Do it.
I have to ask what that
That's a new term for what I believe is called depression. It's basically just hanging out in bed.
Under your blanket. It's, John Lennon and Yoko Ono. They did that for a while as a
Well,
protest.
that was different, though, because they, brought in newspapers and stuff.
Was it?
That was,
And now we bring in laptops and our little devices and watch our Teen Wolf series in bed wrapped by a blanket.
Yeah, that's bad for you. It's bad for you. You need to move around. Bed sores are bad.
Oh, bed sores are terrible. Yeah. I can agree with that one. Okay, let's move on to a few brainy things. We're gonna make you happy and smart before you go home. Maybe give Blair some nightmare juice. Do-do-do-do. Okay, last few stories here.
Thanks.
We love talking about the supportive nature of microglial cells to the neurons in our brains. Once upon a time, it was like, neurons! They're all that our brain needs. They're everything that make us do all the things, and there's nothing else important. Microglia, what is that? That's just packing material, like those styrofoam
tubes or whatever. Yeah.
Oh, not just tubes, but you said tubes. So now I have to follow up on this, which is that researchers studying University of Heidelberg have discovered and they discovered this years ago, but tunneling nanotubes. Microglia connect to your neurons, not just by wrapping around them in a nice hug, but they actually have tubes that go I don't know, like those sky pathways, between skyscrapers where you're like, I'm in one skyscraper on one side of the street, and then I'm gonna walk across this tube to a skyscraper on the other side of the street. The microglia do this with our neurons. Once upon a time, it was this idea that our neurons are just like, boop, neurons. Santiago Ramon y Cajal, and Golgi had these different ideas about how the brain was put together. And then it was like, oh no, finally we discovered it's just like synapses, and they never really connect with each other. They just kind of barely touch, and they have synapses, and that's this thing, right? And that's what we've been talking about. But now we know there are tubes. There are little nanotunnels in our brain between the microglia and the neurons. Why are they there? And so these researchers were like, we think that these tunneling nanotubes are there to help remove waste from the neurons. That this is the garbage conveyor belt. That these microglia have these extensions that are like needles into the neurons that then pull stuff out. That stuff can be transported along. Nobody had any research supporting this or denying this. And so these researchers have just published their study in a journal, Cell Death, and disease. And their most recent study, cell death and disease. And they have really uncovered this protective role for microglia. And the research that they did, they showed that the glia with these nanotubes, first they took neurons and microglia, and they showed that they could put them together in a petri dish and they would grow together, and that these nanotubules were there, and they were nice little nanotubules. And then they were like, okay, so what do cells like to get rid of? How about alpha-synuclein, which is one of those nasty proteins that gums up the works and causes Alzheimer's. So they're like, okay, we're going to make the neurons with alpha-synuclein and then add the microglial cells and see what happens. And they showed that all of the alpha-synuclein went from the neurons down the down those nanotubules into the microglia. And they did it the other way as well. They grew the microglia with the alpha-synuclein and then added the neurons to see if it pushed, just was like a back and forth kind of thing. And it wasn't. The alpha-synuclein preferentially was taken from the neurons into the microglia down these microtubules or nanotubules.
So is there any suggestion that the Alzheimer's is then a microglial needle tube problem?
Yes. And so there has been research, and they also showed that there were that sick neurons, that the microglia would send mitochondria down the nanotubules to the sick neurons, because mitochondria are effective in helping the metabolism of a neuron, so it could fix it up. And so, yes, number of experiments that they did. And this also falls in line with a whole bunch of research that suggests that a lot of neurodegenerative disease has to do with this kind of waste management problem, which would be the microglia, could be something related to proteins or signaling mechanisms that are involved in that transport process down the tubules, whether the nanotubules get formed in the first place, whether they're broken, what happens to upkeep those tubules or not. And so this is a whole new area for researchers to look into and ask a whole bunch of questions, because we have seen that microglia do seem to be implicated in a lot of neurodegenerative diseases. This waste management problem is an issue. Mitochondria, also an issue. So how does this all play together? And this is like a whole new structural issue that now can be studied.
Yeah. It's always also amazing when there's something like this where here's this very key important aspect of a lot of diseases that you've been studying for the last 20, 30 years that you were missing.
Yes.
In all of that research, which a lot of work and smart people and effort has gone in.
But you wouldn't be where you are right now without all that previous research.
Absolutely.
But still,
Well, maybe not all of it, but yeah. We can say that. I give everybody a pat on the back. But that missing component, then ties together probably a bunch of research with and we're scratching our head. We don't know why we see this effect or that effect or how this is involved. And now suddenly there's this key component.
Yeah. Like how have microglia been involved in waste management? How does that work? Oh, well, it's a series of tubes. Oh my gosh, you have tunnels in your brain, nano tunnels in your brain, and they're very important. This next study is related to our diets and aging, and Blair's hopped off for just a second here, but researchers publishing in Science this last week have published a study called Taurine Deficiency as a Driver of Aging. Researchers have shown over the years that taurine is one of those semi-essential amino acids that declines with age. So in our blood, in our urine, the amounts of it decline over time as we age. Yet we know during development, taurine is very important for vision, for neurodevelopment, for musculature development, all sorts of stuff. But then it starts to decline. And so researchers in this paper, they showed that supplementation in mice with taurine slowed a lot of the markers of aging, like DNA damage, telomerase deficiency, impaired mitochondrial function, cellular senescence. We know that humans have a loss of taurine as we get older. And so the idea is that knowing that in mice, and they also looked at it in monkeys, that in these other organisms, that taurine seems to benefit as you get older, even though it's naturally declining. Could we supplement as we get older, and would that be beneficial? It was beneficial to their study animals. So
I just went online and found and ordered
taurine,
a whole bunch of taurine supplements of different description.
5-hour energy. Try not to do the stuff that's got the sugar and the other things that are bad for you, but...
Well, it's supplements, so it's all unregulated, so I have no idea what I'm actually buying or what's going to come in the pill that I'm going to take.
That should be fine.
But I just ordered a bunch so that I can be younger.
So taurine is also found in food. So you can get food. Taurine is in shellfish. It is in meat.
That's going to kill you quick.
Very, very high levels of taurine in organ meat, like liver. Lots of taurine in eggs, but cysteine is one of the precursors of taurine. So there are lots of foods that like vegetarians can eat that aren't necessarily rich in taurine, but they are rich in the precursors to taurine. So perhaps, eating some of these other foods can that are precursor rich can lead you to also increasing your taurine levels. But
did you say eggs? Eggs is on the list, though. That's such an easy way to see. Just do eggs.
It's so easy.
The perfect food.
Yeah, eggs is easy. But yeah, there are many foods out there. If you do some good research and figure it out, take care of yourself, people. Taurine. If your body's naturally declining and how it holds on to it, let's maybe give our bodies a little bit more. Super easy. Super easy.
It is. Let's face it. We don't have the taurine we used to have. It's just...
No, I don't. But we might have new robots for space. And Blair, again, for some reason, I really like ending the show with stories that are going to give Blair some kind of nightmare juice.
Yay!
I want to give you good advice for aging because I want to help you as you grow as a human and grow other humans. But then I also like to terrify you a little bit. Got to balance it. And so researchers from EPFL have published in Nature Machine Intelligence their new modular robot, which is based on inspiration from polygon meshing and swarm behavior.
I don't like the sound of that.
Swarm behavior is always so. It's almost like dehumanizing robots in a way that's like
turning robots into everyone's favorite thing, wasps.
Yeah, so this robot
de-anthropomorphizing or something.
This robot is not very anthropomorphic at all. This is a polygonic triangle.
Looks like a triangular Roomba.
It is like a little triangular Roomba. It's called Mori, and Mori can connect with other Mori units to be able to make a big Mori unit. And so that's the swarm behavior, is that for whatever need that may occur, a bunch of these little triangular Roombas can connect together and be like, we're Voltron, and suddenly they can Yes, and they can maneuver and work as a whole, or they can work as individuals. And so that is part of the interesting aspect of the swarm research that's occurred over the years related to robotics. So in its modular nature, they've been able to show that the modules can come together in such a way to create more complex robots, like a quadrupedal robot that could walk around on, each of the little triangle Roombas could become one of those things is a leg, and they could walk around. They also have ideas that this potentially could be used in like a space station or the International Space Station, even though that'll be probably decommissioned by the time these robots are ready. But the idea is that because of the ways that they can pretty much connect in any configuration, that they could be used for multiple uses, that these robots could create satellite sheets, that they could be used for maneuver by astronauts in space for maneuvering objects around. They could be used on the outside of a space station. They would be small and easy to transport, and their modularity would allow them that kind of eternal efficiency. They're not just a one-use robot. And they wouldn't be able to take over some of the uses that very specialized robots would be good at, but in the end, Maury would be modular and maneuverable and usable for all sorts of things, just what the astronauts need. What?
They are lying to you. This is a macro version of something they are going to continually scale down until you have what is essentially interconnecting nanobots.
Yes.
That act like a liquid and can form anything. That's where this is going.
I don't doubt you, actually.
That's bad. Like, it's in theory, I'm like, yes, that would be great. That would be super beneficial technology. There's lots of cool things about that.
Yeah.
The weaponization of that is bad.
But even nano, it's not weaponization.
So it's everything. Look, here's
two
the
steps
thing.
away from it, and those 2 steps are very easy to do.
This is like the AI disclaimer I gave in the beginning, right?
Yeah.
Anything in the hands of science creates something really cool.
Yes.
You take that same cool thing, you put it in the hands of governments, and it can be a thing that kills.
Yes.
And you can put it in the hands of a corporate environment, and it's something that is used to gain leverage over other humans and take their resources from them. So it just depends on who's using a technology, not what the technology is.
Yes. No, you're correct.
What if it's being used by Felix? What will he do with it?
Right now,
he's blowing bubbles, I saw.
He's blowing bubbles. He's taking all of his belongings and putting them on the balcony.
Good.
They get pushed off. Bye-bye.
He's moving outside to the balcony.
Sorting. That's like a normal thing, right?
Gravity is cool.
Anyway, it's all lies. They're not going to use these giant triangles on the space station. They're going to turn this into nanotechnology so they can have like a fluidic
oh, I love you. Okay.
No, that's 100% what this is. It's
okay. All right, Maury 3 creators. We know what you're up to now.
Yeah. You're not going to use those giant triangles? No.
The researchers say these robots, we had to rethink the way we understand robotics. These robots can change their own shape, attach to each other, communicate, and reconfigure to form functional and articulated structures. So yeah, they're going to be great.
It's very cool. It has the potential to be very cool.
You just, go ahead and go to sleep with that in your mind.
It's all right.
Did we finish the show? Are we done?
What we really need them to do is replace humans, so humans don't have to go to space anymore.
Or so that we don't mess up our own planet anymore. But anyway, I like being human. I don't want to be taken over by AIs or robots.
Unless they're in the right hands. See, this is the thing.
Whose hands? Are the
about
robots' hands or human
Justin's.
hands?
Obviously, Justin means him.
Shark hands?
Well,
what are you talking about?
what I mean is that, like, we live on a planet that is half authoritarian government, right? We live in a place where
Very
there are
democratic.
malicious humans who are in control of resources and things. Like, there's one level where, like, is the AI robot overlords really going to be worse than what we've already got?
Or are we going to train
better.
them on what currently exists and model bad behavior and make things worse?
Didn't you see the study that was out this last week? AI being trained on AI or machine learning being trained on machine learning for all these chat GPT uses and how it's accelerating the decline of the intelligence of the chat of the machine.
That's
The
great.
same
I love
thing
the idea
happens
it's
with-
just going to self-implode and we're going to have to start over.
We're gonna start over. There you go. It's gonna be so good.
I actually did see an AI researcher mention that. Like, this isn't going to go anywhere. This is all going to implode and we're going to have to start over at square one. Like, all right.
We'll see. Oh, the future. Who are we to tell the future?
Just don't hand those triangles AI, please. Not yet.
What was the Gravity Falls triangle?
Bill.
They're
Mhm.
they're like modular bills. Bill's
Oh God, that's not good. We don't need a billion tiny Bill ciphers.
No, we do not. No, we do not.
Okay.
All right, we made it to the end of the show. Yeah.
We sure did. Yes.
Oh. Well, everyone, I want to say thank you for joining us for another episode. Everyone in the chat room, thank you so much for chatting. Discord, thank you for being in the Discord with all your comments and chats. You know, if you're a patron, then you can join our Discord. Another reason to join our Patreon. But I do want to say thank you also to Fada for help with social media and show notes, and to Identity4 for recording the show, to Gord and Arnlore and everyone who hangs out in the chat and makes sure that everybody's being nice to everybody else, and to all of our Patreon sponsors. Thank you to Craig Potts, Marie Gertz, Teresa Smith, Richard Badge, Kent Northcote, Rick Loveman, George Corris, Pierre Velazarb, John Ratnaswamy, Carl Kornfeld, Chris Wozniak, Vegard Shefstad, Hal Snyder, Donathan Styles, a.k.a. Don Stylo, Ali Coffin, Reagan, Don Mundus, P.I.G., Stephen Oliveran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Kevin Reardon Noodles, Jack Bryan, Carrington, David E. Youngblood, Sean Clarence, Lamb, John McKee, Greg Riley, Mark Hessenflow, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minnis, Johnny Gridley, Remy Day, Flying Ulrich, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Deb Wilkinson, Rodney Lewis, Paul, Rick Ramos, Philip Shane, Kurt Larson, Craig Landon, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Adam Mishkan, Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, and Tony Steele. Thank you so much for all of your support on Patreon. And if you would like to support us on Patreon, head over to twist.org and click on that Patreon link. And I'm just realizing that I forgot to thank Rachel for her help editing the show.
Oh, yes, of course.
Yeah. And
Rachel,
will
do us a favor, cut that out, edit it, and put it in where it belonged before.
Put it at the beginning of the show.
Thank you.
Put it right at the beginning of the show.
Word all
We'll open with
below.
it.
Yes. And on next week's show,
We will be back broadcasting on Wednesday at 8 p.m. Pacific time and again Thursday, 5 a.m. Central European time from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Perhaps while you count how many microplastics are in the sand in your sandbox? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org, where you can read about, you can listen to past episodes there, you can read up on show notes from past episodes, and you can also sign up for a newsletter one day.
You can also contact us directly. Email Kiki at Kirsten@thisweekinscience.com. Justin at twistmenina@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be absorbed by a conglomeration of tiny triangle robots that will read it themselves, but not deliver it back to us because they have not been trained to do so.
Yes, and you can hit us up in the meantime on Twitter, where we might not respond because we might not be checking it anymore. I'm not really sure.
My Twitter's broken,
happen?
so
I go there and I check it sometimes, and then I'm like just sad about humanity, and then I leave.
It's just an awful feed now. But,
they won't let me tweet, so I've given up.
So use the emails that Blair was talking about, because we love your feedback, and we do want to cover subjects and topics you are interested in. So if there's a haiku that comes through the night or a story that you think we've missed, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.